Hi, today we're going to cover chapter 14 where we talk about Mendel and genetics. You may be familiar with this from a previous course. We are just going to do it at a cursory level, looking at simple basic genetics to give you an idea of what exists. So why do you look like your parents? That's the question that we want to think about here. Classically, the idea was that you were a blend. If dad was really tall and mom was really short, you'd be somewhere in between. Another idea was a particulate inheritance pattern, that you inherited a particle of something from either parent. And this is Mendel right here, who in a very systematic way showed us that inheritance is a particulate inheritance. Mendel worked with pea plants. This was a really good choice for him because as he looked at inheritance patterns, one advantage was that there were lots of varieties. So he could go to Home Depot or Gibo's or the local seed store and he could buy peas that were purple or white or green or yellow. They have a short generation time, so he could easily get offspring. You could get lots of offspring, and it was easy to control mating. He did this with the following technique. So the flower is the reproductive organ of the plant, and he would choose who mom was going to be. In this case, purple flower is going to be mom. So what he would do is he would cut out all of the male parts, leaving only the female parts. So now we know for sure that this is mom. Then for men, for dad, what he would do is he would go and pick up the sperm, which is the pollen, and he would brush pollen from the white flower on the purple flower. We know exactly who mom and dad were. He allowed them to have babies. To see what the babies looked like, he would then plant them in the ground and look at the offspring. In a typical experiment, Mendel would make two contrasting true breeding varieties. This is called hybridization. So he would take a purple flower, time a white flower, and see what they looked like. The original generation is called the P generation. It stands for parental, not for P as in pea plants. That first generation is called the F1 generation. These are hybrids. And then from there, he went to a second generation. You can think of these as grandkids, where he would start to see something called a 3-to-1 ratio. Here's a typical experiment. So in the P generation, he takes true breeding plants. True breeding plants means that he was guaranteed from the seed factory that they would always be able to produce that particular offspring. So if you're a purple flower, true breeding plant, you're always going to have purple offspring. If you're a white flower that's true breeding, you're always going to have white flowers. So he took these two P generation plants, he calls them, he gets the F1 hybrids. In the F1 generation, all the plants have purple flowers. He then allows these F1 hybrids to self or cross-pollinate. So he's allowing this purple plant right here to cross with itself. And he goes out a second generation. And in that second generation, he sees that he gets purple plants again, but he does get a few white ones back. He gets about 3 purples to every 1 white plant. And what he notices is that the purple color seems to be more dominant. It seems to be stronger. When it's purple times white, all the babies are purple. When you take these purple F1 hybrids and cross, you get 3 purples to every 1 white flower. He does not understand genetics the same way we did. He does not have the vocabulary of alleles and DNA and genes. So his explanations are sometimes a little bit funny, but we want to take what he learned and apply it to our current understanding of DNA, mitosis, and meiosis. So what he did was he said that purple is a dominant trait. He actually called it a purple flower factor. This is what we would now call a gene. But we're going to just say purple is a dominant trait. By convention, we use a single letter to talk about a trait. So in this case, he's talking about flower color. So we're going to use a single color to talk about flower color. Note that we're using the uppercase letter P because it's dominant. White is a recessive trait. We're using a lowercase letter to represent it. Notice that we're still sticking with the letter P because we chose to use the letter P for this entire trait. He saw this 3-to-1 pattern though in other characteristics, and I want you to just look at this for a moment. So over here on the side, when he did his purple to white cross, he didn't just get 3 purples to 1 white flower. No, he planted and grew and watered and counted 705 purple plants to 224 white flowered plants to get that 3-to-1 ratio. And again, he doesn't stop there. He does this again. He does it with yellow versus green seeds, and he gets over 6,000 yellow seeds to the 2,000 green seeds. I have never counted 6,000 of anything in my life. I mean, that's just unstoppable, right? He just sees this pattern over and over and over again, and he continues. He looks at seed shape, he looks at pod color, pod shape, flower position, stem length. He looks over and over at these traits, and he's over and over seeing this 3-to-1 ratio. So he has some hypotheses that help explain what we are observing there in the F2 generation. Alternative versions of genes account for variations. So as a reminder, genes are generic categories. So flower color, that's a gene. There are alternative versions of genes called alleles. You've got the purple allele or the white allele. Each gene is going to reside at a very specific locus. Down here at the bottom, we can see this. So we've got our gene for flower color. It lives at that particular spot on those homologous chromosomes. And then we can see there are two different alleles. In this case, the blue chromosome has the allele for purple flower color, whereas the red chromosome has the allele for white flower color. And again, to take this and expand upon what we understand, what does this mean? At the DNA level, there is a difference happening. So if we look at the gene for flower coloration, in the individual that has the purple flower gene or allele, allele, their DNA is going to go through gene expression and make a protein that codes for purple flower color. What's happening to an individual that inherits the allele for the white flower coloration? Well, their DNA has been mutated. It's slightly different, and so they're not making a protein. And so these alleles are linked with what we've been learning about gene expression, transcription, and translation. An organism is going to have two alleles for each trait. They're going to get an allele from their mom and an allele from their dad. If they have two of the same trait, we say that they are homozygous. You can be either homozygous dominant or homozygous recessive. If an individual is heterozygous, they have two different alleles for a trait. So you can be big P, we would say that that individual is homozygous dominant, little p, homozygous recessive. If you happen to be heterozygous, the heterozygous individual will have the appearance of whatever the dominant allele is. Lastly, the law of segregation. The law of segregation says that two alleles for a heritable character will separate or segregate during gamete formation. What occurs is that the individual is going to be able to take those two alleles and separate them into two different gametes. This is sometimes so simple that it becomes complicated, but we're saying the following. If you're the individual big P, little p, and you go to make eggs, half of your eggs will have that big P in them, and the other half of your eggs will have the little p in them. That's the law of segregation. The idea that these alleles are separating when we make gametes. The possible combination of sperm and egg can be shown using a Punnett square. You are going to be asked on your final exam how to solve genetics words problems. One type of genetics word problem that you're going to have to solve is called a monohybrid cross. I will start with that type of cross and show you how to solve them, and then we'll talk about additional types of crosses where you'll use these same steps as well. So to solve the genetics word problems, the first thing that you need to do is you need to write down the genotypes of the parents. Or in other words, you need to take the story that says tall plant or short plant or red plant or polka dotted plant and turn that into letters. Secondly, you'll figure out the gametes that each parent can produce using the law of segregation. Then you'll place those gametes on the outside of a Punnett square, and then lastly, you fill in the boxes. We can go ahead and look through this step as we look at our original Mendelian cross. So we had the purple times the white flower in the P generation, and in the F1 generation, we had all purple hybrids. They were all heterozygous purple. Each of these purple flowers had gotten one dominant allele from dad and one recessive allele from mom, so they were all big P, little p. And so what we did was in the F1 generation, we allowed this cross to cross with itself. So big P times big P, little p. Those heterozygous purple plants crossed. In this case, what we will do is we'll take each individual and figure out what gametes they can produce and place those on the outside of a box. So individual number 1 can make big P and little p gametes. Individual 2 can make big P and little p gametes. So here this is much prettier. Big P and little p from one parent, big P and little p from the other parent. And then we fill in the boxes. As we fill in the boxes, we see our 3 purple to every 1 white plant. At this point, it's really important to emphasize some useful genetic vocabulary. When we've been talking about 3 purples to 1 white plant, we've been talking about phenotype. Phenotype is the physical appearance. You look purple, you look white. A related term, although somewhat different, is something known as genotype. Genotype is the genetic makeup. If we look at our original cross, we actually had 1 homozygous dominant purple plant, 2 heterozygous purple plants, and 1 homozygous recessive white plant. This trait of 1 to 2 to 1 is a genotypic ratio, where we're talking about the genotypes of the plants. By convention, we always talk about the homozygous dominant first, then the heterozygotes, then the homozygous recessive. And in this case, we see a 1 to 2 to 1 genetic ratio. Here we have a sample monohybrid cross, something similar to what you might see on a test, to work through to practice this step. So in the story problem, I will always let you know what's dominant and what's recessive. You don't have to memorize a special list. So here's how it starts. Red is dominant to orange. A heterozygous red flower mates with an orange flower. What's the probability of having red flower offspring? Okay, so remember, step 1 is to figure out the genotypes of the parents. We've got to write down the letters. We've got to change this sentence into into actual letters we can use. So I mentioned that red is dominant to orange, so we're going to choose a single letter to represent color. I'm going to choose letter R because my letter R's often look different, and I'm just going to give myself a little bit of a reminder that big R means red and little r means orange. So let's look at the first parent now. A heterozygous red flower. Heterozygous means 2 different alleles. So heterozygous red flower would appear big R, little r. And then the second parent, if we look at the second parent, they are an orange flower. It doesn't say homozygous or heterozygous, but I am able to figure out what an orange flower would be because this is the recessive trait. So an orange flowered individual would look like, little r, little r. So now I've done step 1. I figured out the genotypes of both parents. Step number 2, I need to figure out what kinds of gametes these individuals can produce. So my heterozygous red flower can produce big R gametes or little r gametes, whereas my orange flower individual can produce little r gametes or little r gametes. Now that I know the gametes that each individual can possess, I can go ahead and set up my Punnett square. My Punnett square is going to be a 2 by 2 grid because I have 2 gametes that one individual can make multiplied by 2 gametes that the other individual can make. So I'm going to set up my square. It's a 2 by 2 square. I'm going to put one individual across one side, big R, little r, and then the other individual across the other side. At this point, I'm now going to just fill in each box. I'm just going to pull in the 2 letters and write them together. By convention, we usually use the uppercase letter in front of the lowercase letter. That'll just help you scan these better. We also keep genes together. So big R, little r, big R, little r, little r, little r, little r, little r. We can now go ahead and go back to our question and now answer what was originally asked. What is the probability of having a red flower offspring? So I look at my Punnett square. This individual is going to be red. This individual is going to be red. This individual is going to be orange. This individual is going to be orange. So the answer to the question, what is the probability in this cross of having a red flower offspring? The answer would be 50%, or you could say 2 out of 4, or you could say 1 out of 2. All of those would be correct answers. Now that we've seen a monohybrid cross, we want to talk about another type of cross called a test cross. This is another type of cross that is guaranteed to be on your final exam. In a test cross, what you're trying to do is you're trying to figure out the genotype of a particular organism. So for instance, I've got this beautiful picture of a purple flower right here. Here's a purple pea plant, and I ask you, what is the genotype of this purple flower? And you look at me and you say, I have no idea, because this purple flower, it could be either homozygous dominant purple, or maybe it's heterozygous, right? Either of those options could be the potential genotype for this flower. So in a test cross, what you do is you take your mystery individual and you breed that mystery individual with a homozygous recessive individual. So here it is written out. You take your mystery individual and you breed them with a homozygous recessive individual, and then you look at the offspring to see what happens. Here are your potential options. So here's my mystery individual, and I mated them with a homozygous recessive individual. In one option, going here towards the left, if your mystery individual was a homozygous dominant individual, if they happen to be this, when you do the cross, all of their offspring will be purple. So if you do the test cross and you get all of the offspring being that dominant phenotype, it's highly likely that this individual is also homozygous dominant. If, however, when you do the test cross, your mystery individual was a heterozygote, now going towards the right-hand side of this picture. If your individual was a heterozygote, when you cross them, you're going to see about a 1-to-1 ratio, or a 50-50 split. About half the offspring will be purple and about half the offspring will be white. You should be able to analyze the results of a test cross and tell me what the results mean. So again, if you see that sort of 50-50 result, you know that that mystery individual that you started off with had to have been heterozygous. You should also know what a test cross is. It's where you take a mystery individual and mate them with a homozygous recessive. Here's a great example for you to sort of practice this with. Tongue curling, or taco tongue, right, is one of the very few human traits that's a simple dominant recessive thing. Big T means yes, you can curl your tongue and do taco tongue. Little t means you can't curl your tongue. So this is our family from a long time ago. This right here, Dr. Miller, if we look at him, he's a great guy, but he cannot curl his tongue. So his genotype is little t, little t. Okay. And there's me, and you know that I can curl my tongue. And so you know that I'm big T, but you're not sure about the other part. Am I big T, or am I big T, little t? I don't know. So this turns out to be a test cross, right? Mystery individual me times a homozygous recessive, and then we look at the offspring and see what they can do. So I'll give you some examples of what can happen. Samantha is totally able to curl her tongue. She's just being a goobah. So yes, she can curl her tongue. Joshua can also curl his tongue. Nathan the baby, he cannot curl his tongue. He is little t, little t. So given these results, what's my genotype? Hopefully that should be pretty straightforward to see, is that Nathan must have gotten one of those little t alleles from dad, but that other little t allele had to come from mom. So what is my exact genotype? My exact genotype is I must be heterozygous for tongue curling. Again, this is just an example of a test cross. Next that we want to talk about is called the law of independent assortment. So far, we've just been looking at monohybrid crosses where we look at a single characteristic. We now want to look at a dihybrid cross where what Mendel did was he looked at two characteristics. His question that he wanted to answer was, are characteristics going to be inherited together as a package deal, or are they going to be inherited separately? So what he did was he looked at two traits. One was looking at whether seeds were round or whether they were wrinkled. Okay, so that's one trait. The other trait that he was looking at is coloration. We're really used to seeing green pea plants. However, there are also yellow seeds that can be produced. So yellow versus green. And this was his question. Are these characteristics going to be inherited together or separately? Let me try to talk you through what Mendel did in his experiment. Through his crosses of individual traits, he already knew the following. He knew that yellow is dominant to green. He uses a big Y to represent yellow and a little y to represent green. And he already knew that round is dominant to wrinkled. He's going to use a big letter R to represent round and a little letter r to represent wrinkled. And what he did in this first case is in his parental generation, he took a totally yellow round plant. So yellow round, that dominant trait, and he crossed it with a green wrinkled plant. And then he got his F1 generation. In his F1 generation, all of the organisms looked like the following. They had a big Y from dad and a little y from mom, and a big R from dad and a little r from mom. They were all yellow round hybrids. He then allowed these F1 hybrids to cross, and he looked to see what happened. And what he wanted to know is the following. He was trying to figure out, is this a package deal? Because big Y and big R were stuck together at the beginning, are they somehow still stuck together right now? And as an organism, if they get big Y, do they always have to also get big R at the same time? Similarly, are little y and little r somehow stuck together? If a person gets little y, do they have to get little r at the same time? When he did the cross, what he noticed is that this is not the case. These traits are not inherited together. They're not packaged together. Instead, independent assortment is happening. Independent assortment says that we can have any combination of alleles when we are going through meiosis to make these gametes. I want to show you sort of what's happening in this particular case. So what happens is when we look at these F1 individuals, if we start here with dad, big Y can line up with any of our letter R's. So we have the possibility of getting a gamete that's big Y, big R, or we also have a possibility of getting a gamete that's big Y, little r. Likewise, you have the possibility of getting little y, big R, or little y, little r. Little y, big R, little y, little r. So this individual right here can make this whole large possibilities of gametes because of independent assortment. If we look at the other individual right here, this individual can also make 4 unique gametes. So again, big Y can combine with big R, or big Y can combine with little r, or little y can combine with big R, little y can combine with little r. So this second individual can also make 4 unique gametes. If we were doing the Punnett square for this particular cross, what we would now see is that we would need to use a 4x4 grid to describe what is happening. We would need to put all of the gametes from one individual on one side and all of the gametes from the second individual on the second side. And this is what Mendel saw happening. We're going to go ahead and look at a picture of that from the text. Here you can see a pretty picture from our textbook showing how Mendel worked through this law of independent assortment. Here is the parental generation where he took the yellow round plant times the green wrinkled plant. In the F1 generation, he got all yellow round hybrids. If yellow and round were somehow linked together and green and wrinkled were linked together, he would have expected to see very simplistic results from his cross that looked like this. That's totally wrong though. He did not see those results. Instead, when he took the F1 generation and went to the second generation, what he saw was that these 2 alleles are segregating independently of one another. So he got yellow round seeds, but he also got a brand new combination. He got yellow wrinkled. And yes, he got green wrinkled, but he also got a brand new combination. He also got green round. And he saw that this happened and gave him a ratio of approximately 9 to 3 to 3 to 1. This law of independent assortment should sound very familiar to us. This is what we talked about when we talked about meiosis and genetic variation. Remember, the law of independent assortment says that when we look at homologous chromosomes and the way that they're lining up during meiosis, they can line up in a variety of different ways. They might line up with all the black on one side and all the red on the other side, or they might line up with black and red swapped, right? As long as those homologous chromosomes are segregating, what the next pair of homologous chromosomes does really doesn't matter. And so this is the law of independent assortment. You should be comfortable taking any individual that I give you and figuring out what type of gametes they might produce. So similar to what we just did on the previous page, if you have this individual and you're trying to figure out what gametes they can do, you are looking at every combination of letters Y's and letters R. So you can do FOIL. First letters, big Y, big R. Outer, big Y, little r. Inner, little y, little r. And last, little y, little r. There's your fourth. So you should be able to take any individual, especially somebody who has two traits, and figure out the possible gametes they can produce. Here's an example problem that we can work through to see if we understand how to do this. So I'm going to tell you that red is dominant to white and tall is dominant to short. For simplicity's sake, I'm going to use big R to represent red and little r to represent white. I'm going to use big T to represent tall and little t to represent short. Note that I'm using the same letter for the same trait. So when we're looking at color, I'm just using letter R's. When I'm looking at the height, I'm just using the letters T. All right, so the first thing we need to do is we need to figure out the genotypes of our parents. A heterozygous red, homozygous short plant. That's one individual. So heterozygous red would be big R, little r. Homozygous short would be little t, little t. That's parent number one. Mates with a heterozygous red, heterozygous tall plant. So what does that individual look like? Heterozygous red, heterozygous tall plant. We now have the two individuals, and we need to figure out what possible gametes they could produce. In order to figure out what gametes they can produce, we're going to do FOIL, big R, little t, big R, little t. Little r, little t, little r, little t. So that first individual can produce the following gametes. Note that when I was figuring out what gametes that individual can produce, I was just focusing in on what that individual could produce. I wasn't doing anything with the second individual. I then need to figure out what gametes the second individual can produce. Big R, big T, big R, little t. Little r, big T, little r, little t. So here we go. Big R, big T. Big R, little t. Little r, big T. Little r, little t. I now have this set up so that I can fill out my Punnett square. My Punnett square is going to need to be a 4x4 grid. I can now start by filling in each of these individual squares. Remember that we're going to use uppercase letters in front of lowercase letters. We are also going to keep genes together. So the R's are going to stay clustered together and the T's are going to stay clustered together. This will just help us scan it as we look at it. It's totally okay if you move them around and put them in different places. It just becomes really hard to scan the Punnett square to look for an answer. So starting up here in this corner, we've got big R, big T, little t. In this one, it would be big R, little t, little t. Big R, big T, little t. Big R, little t, little t. And you just keep filling this out as you go along. I'll finish this off. Big R, big R, big T, little t, big R, little t, little t, little r, big T, little t, little r, little t, little t, and then our last row. Now that you have this chart filled out, you would be able to answer a question about the offspring. So I could ask you, how many of their children are going to be red and tall? Or how many of their children are going to be white and short? Let's try that one. How many children are going to be white and short? So in order to be white and short, you would need to be little r, little t, little t. If we look at this, here is our white short individual and our white short individual. So the answer would be 2/16 or 1/8 of the individuals are going to be white and short. And you can set these up. This is going to take a little bit longer time. These are called dihybrid crosses. You will be asked to complete a dihybrid cross on the final exam. One of the nice things about Mendelian inheritance is that the laws of probabilities apply to these types of crosses. One important probability rule is called the multiplication rule. This is the probability that two or more independent events will occur together is the product of their individual probabilities. We can use this handy rule to help us when crosses begin to get very complex. So for a monohybrid cross and a dihybrid cross, it's pretty easy to draw out the Punnett square. However, when we start looking at 3 or 5 or 7 or 9 genes at the same time, our Punnett squares become humongous, and none of us have time to draw a 64 by 64 grid to figure out all the different combinations. Lucky for us, though, we can use probability to answer these types of questions. They're called multi-character crosses. You will be given one of these types of crosses to work on the final as well. So a multi-character cross looks the following way. What we do is we will give you some parents. So here's parent, big A, big B, big C, little c, crosses with another heterozygous A, heterozygous B, heterozygous C individual. And we ask you, what is the probability of getting a very particular child? Although we could set up a Punnett square, it's going to be such a large Punnett square that it's going to be almost impossible to solve. A much quicker way to solve this is to use this multiplication rule. Let's do that on the next slide. What we're going to do is, instead of trying to look at every trait at the same time, we're going to break each trait apart. So let's start by just focusing on the letter A's, right? If I told you to figure out the probability for this particular cross, big A times big A, little a, what's the probability of getting somebody who's big A, you could hopefully do that very quickly by setting up a little monohybrid cross. You run your cross and you find out, oh, it's 1/4 of a probability to get an individual who's big A from that cross. And then you just hold on to that. And now you're going to focus on the next letter. So let's focus on the letter B's. If I have a parent who's big B and a parent who's big B, what is the probability of getting a child who is also big B? And it turns out, as we fill in these squares, the probability here turns out to be 1/2. We then focus on our final value. If you have a parent who's big C and another parent who's big C, you can set up the cross. What is the probability of getting a kid who's also big C? That also happens to be 1/2. So what you've done is you've taken this very complex problem and you've broken it up into individual traits. From here, because of the rules of probability, what we can do is we can multiply all of these numbers together. 1/4 times 1/2 times 1/2 equals 1/16. If it's been a while since you've done multiplication with fractions, it's not as hard as you maybe think. You just go ahead and you multiply across the top, and then you multiply across the bottom. So 1 times 1 equals 1 times 1 equals 1. And then across the bottom, 4 times 2 is 8. 8 times 2 is 16. In these cases, you'll note it's easier if we keep the probabilities as fractions instead of turning them into percentages, right? I can easily do this math if I think 1/4 times 1/2, not 25% times 50%. It gets really sloppy. What we want to do now is we want to start looking at inheritance patterns that are often more complex than what we saw in simple Mendelian monohybrid and dihybrid crosses. First thing we want to mention is that dominant alleles are not necessarily more common than recessive alleles. So I wish we were in class so I could have you guys all show up your hands. I'm assuming that all of you guys have 5 fingers. That means that all of you, including myself, we are all little f, little f. None of you guys are dominant for fingerness. None of you guys are this or this. Why do I know that? It's because if you have the dominant allele for fingerness, you actually end up having 6 fingers or 6 toes. This is one of those cases where this is really rare. So the dominant allele is not the most common allele. The dominant allele is simply the allele that if both alleles are expressed, it is the one expressed over the other. Polydactyly is actually fairly common in the United States. It's in 1 out of every 400 individuals. What happens when an individual is born with polydactyly, usually often really early on at birth, the surgeon will just remove that extra digit. But I think it would be really cool if one of you guys had 6 fingers, so if you do, please definitely send a picture. That's amazing. As we go through our next couple of slides, what I'm going to do is I'm going to describe a bunch of different inheritance patterns that you should be able to recognize. You're not going to need to run Punnett squares for these crosses, but you should be able to recognize them and predict what's going to happen. So the first one is complete dominance. This is what we've been talking about this entire lecture. This is a monohybrid cross, a dihybrid cross, the test cross, the multi-character cross. There's this different thing though called incomplete dominance. In incomplete dominance, neither trait is completely dominant over the other. This is going to be, you've got a red flower and a white flower and their babies are pink, right? Neither is completely dominant. Or you have a yellow flower and a red flower and their babies are orange, right? Neither is completely dominant. Another example that we're going to talk about is something called codominance. Codominance is when 2 alleles get expressed at the exact same time, so they're both there. This would be purple times a white flower, and you get purple and white polka dots. Or a red flower times a white flower, and now you get red and white stripes. As we look at codominance, a very specific example that we'll look at is specifically AB blood. So not all bloods, but if you happen to be type AB blood, you have a type of blood that's presenting both A antigens and B antigens. So both they're there at the same time. Here's an example that we'll work through. As we work through, try to think which type of inheritance is she showing? Is she showing regular complete dominance? Is this incomplete dominance, or is this codominance? So we've got our red flower and our white flower. They have babies. In the F1 generation, they're all pink. And in the F2 generation, we see reds and pinks and whites. Hopefully you recognize that this is an example of incomplete dominance. Neither red nor white was completely dominant, and so we got this intermediate hybrid. Notice that this isn't blending. Those particles still exist. We can go back to having reds and whites in that second generation. Another inheritance pattern that we can talk about is called multiple alleles. Many genes have more than 2 alleles. Right now we've been keeping things very simple. We've been saying, oh, there's a big T or a little t. You can be tall or you can be short. There's just 2 options. But for most genes, there's way more than just 2 options. Like even if we think about something for hair color, it's not you have black hair or you have blonde hair. No, there's black hair and there's brown hair and there's blonde hair and there's red hair and there's gray hair, right? There's all sorts of different options. So this is called multiple alleles. Most genes have more than just 2 alleles. A simple case of this is with blood type. When it comes to determining your blood type, you are only going to have 2 alleles, right? Every individual is just going to have 2 alleles, but there's 3 options to choose from. So there's a dominant allele called the A allele. You notice that we're using an uppercase letter I to represent that this is the dominant allele. This is going to code for the placement of A sugars or A antigens on the surface of the blood. There is a dominant allele called the B allele, uppercase letter I with that B superscript. This is going to code for the placement of B sugars or B antigens on the surface of the blood. And then there's a recessive allele, little letter I, that codes for the placement of no antigens on the surface of the blood. Again, we've got 3 options. So this is an example of multiple alleles. As those alleles come together, this gives us various blood types. For instance, let's just go through a couple of them. If you're this individual, what type of blood would this individual have? This individual would have type A blood because they have that A antigen expressed, even though they have that recessive allele. We're not talking about positive and negative at this time. That's another allele that we would need to look at, and that would turn into a dihybrid cross. So we're just looking at A, B, O, that type of stuff right now. Let's try this individual. If you're this individual, what kind of blood would you have? This individual would have type B blood. This individual right here, if you have 2 recessive alleles, this is what's meant by type O blood. It means that there are no antigens on the surface of the blood. And then this individual, they have both A and B antigens expressed at the same time. So this is an example of codominance, right? Where they've got the A and the B antigen present at the same time. Here's a nice picture of this from your textbook. You can see if you have that dominant A allele, then you end up with the A antigens on the surface of your blood. If you have the dominant B allele, then you end up with the B antigens on the surface of your blood. If you have both the A and the B allele, you're going to end up with both antigens on the surface of your blood. And then somebody who has the recessive allele does not have any antigens on the surface of their blood. You should be comfortable with knowing how blood typing works and what it means if somebody's type A or type B or I or AB blood. Sorry, O or AB blood. The next type of inheritance that we want to talk about is called pleiotropy. Pleiotropy is the situation where one gene will influence many traits. For example, cystic fibrosis or sickle cell disease. We've looked at these cases before. In both of these, there's one problem in the DNA. Right? One small mutation that happens at the DNA level that causes a mutation in the protein. And because of that one small problem, multiple impacts are found in the individual. So somebody with sickle cell disease, they're going to probably have organ damage. They're going to have jaundice. They're going to have painful muscle aches. They may have liver damage. They may have organ damage, right? All of those bad things are traced back to just one gene. So you've got this one gene influencing many traits, and this is called pleiotropy, where one gene influences many traits. In epistasis, this is a really fun example of where a gene at one place is going to influence how a second gene is phenotypically expressed. The example that I'll always use when talking about this is for mammalian coat color. So let's just talk about labs for a moment. So Labrador retrievers, they have a gene that determines the pigment color. It turns out that big B means that they'll make a black pigment, and little b means that they make a brown pigment. So that's one gene. Big B means you're black, little b means you're going to be brown. Okay. There's a second gene that's going on. There's a second gene that we're going to use the letter E to describe. Letter E means yes, place the pigment down. Lowercase letter e means don't put the pigment down. And so these two genes are going to interact. The gene that codes for whether or not we deposit the pigment, the letter E gene, is going to alter the phenotypic expression of the second gene, the letter B gene. Let's look at an example. Here we've got 2 dogs, and you can see their offspring, and let's just go through some of them and make sense of it. So let's start up here. This dog right here, big B means you're black. Big E means you deposit the black color down. Yep, that dog is black. That makes sense. All right. What about this individual right here? Little b. What does that mean? It means they're brown. Big E. Oh, you deposit the brown. Oh, this individual is brown. What about this dog right here? What does big B, little b mean? Big B, little b means that this dog has the ability to make the black pigment. But why aren't they black? Oh, it's that letter E, right? The letter E is saying don't put the pigment down. And so this is where we get our golden lab. Likewise, here we've got this individual. It says little b, they should be brown, but they're not because that letter E is influencing the letter B. So you've got a gene at one position altering the phenotypic expression of a gene at a second position. And again, mammalian coat color is a great example of this. Now I want to talk about something called polygenic inheritance. Polygenic inheritance should not be confused with pleiotropy. So let's talk polygenic. Poly means many, genic means genes. In polygenic inheritance, you are going to see that many genes are going to influence a particular characteristic. So it's almost the inverse of pleiotropy. Earlier, we talked about pleiotropy. That was one gene, lots of traits. Polygenic says, no, we're going to have lots of genes that influence just one trait. Most human characteristics that you would have questions about turn out to be polygenically inherited. So when we talk about your skin tone or your height or your eye color or whether or not you're going to have cancer or whether or not you're going to get heart attacks, all of those are the result of lots and lots of genes influencing that single trait. Here's a very simplistic example of how skin color may work. In this example, we are assuming that skin color is determined by just 3 genes. It's actually much more complicated than this, but we're going to just use this as an example. And what we're going to start with is up at the top, sort of 2 intermediate skin-toned individuals. Both mom and dad sort of have intermediate skin tone. They have a couple of light alleles and a couple of dark alleles. If we look at this Punnett square, what this is showing is it's showing us the possible children that they may have. So most of their children will have intermediate skin tone just like mom and dad. But it's possible that one of their kids could get every single dark allele from dad and every single dark allele from mom and have really dark colored skin. Likewise, they could have a kid that got every single light allele from mom and most of the light alleles from dad, and they have really light colored skin. You've probably seen this in your families. You've got that brother or that uncle that looks Italian even though nobody else is Italian. They are your family members, right? This is just an example of polygenic inheritance where you've got lots of genes influencing this single trait. Here's an example of polygenic inheritance, again, thinking about eye color. So this is our family, our eye color. Adam has just pretty normal brown eyes. I've got brown eyes. Samantha has brown eyes. There's Nathan's light eyes, and then Joshua's really dark eyes. How does this work? It's not big B means you're brown and little b means you're blue. It's more complicated than that. Eye color is multiple genes that are influencing this trait. So what happens in Samantha here, she gets a couple of dark ones and a couple of light ones from mom and dad, and she ends up with nice brown eyes. How does Nathan end up with such light eyes? He gets every single light allele from mom, every single light allele from dad, and he ends up with really light colored eyes. And then Joshua, how does he end up with these really dark colored eyes? Gets all the dark alleles from mom, all the dark alleles from dad, and he has really dark colored eyes. Please don't believe what you've seen on every episode of Law and Order or whatever crime scenes show you've ever watched. Eye color is a polygenic inherited trait. There are many genes that influence eye color. It's not simply a dominant recessive, big B you're this, little b you're that. There's lots of genes that are influencing eye color. So we've been through a lot of examples now that have talked through inheritance. We looked at complete dominance, incomplete dominance, codominance, multiple alleles, pleiotropy, epistasis, polygenic inheritance. All of those examples, we could still try to describe what was going on using Punnett squares. Important though to mention is that what you look like really depends on the environment. Organisms are not just responding to things genetically, they are responding to things environmentally. These 2 plants down here, they're called hydrangea plants. You might have seen them before. They get planted here in the south. These 2 plants genetically are exactly the same. They're both big A, big A, big A, big A, right? Genetically, they're the same. But one's obviously pink and the other one's blue. What's going on? This is all a difference of environment. Depending on where you plant the plant, whether it's in a basic or acidic soil, you'll get different coloration. So although it's fun as a biologist to think about Punnett squares and genetics crosses, it's really important to remember that the environment has a major impact on what the phenotype of the organism is eventually going to look like. Multifactorial characteristics are those that have both genetic and environmental influences. This is arms of your textbook author and then her husband and her kids. And if we just think about this from an environmental standpoint, why is mom so pale and the kids are tanner? Well, I bet the kids still go to recess, and I bet mom's inside at work all day. So we're not talking about genes, we're just talking about the environment. Again, the environment, most of the times, I would say is at least 50% of the story, whereas the genetics are the other 50% of the story. Here's a great example of this happening in monkeys. And what they found is that there is a gene, and this gene would get expressed differently based on where they were dominant or not dominant. So if they were the alpha male, right, if they really have this high position of social hierarchy, this gene expression is different than if you've got somebody who's at the bottom of the dominant hierarchy, and then their DNA expression is changing. So this is all environmentally induced, right? The gene is there, but whether or not it is expressed is an impact of this hierarchy. Here's another example. These are two identical twins that unfortunately were separated and fostered out in two distinct homes. One was raised in a real nurturing environment, and the other unfortunately went through a lot of abuse and neglect. And you can see these are identical twins. Their DNA is exactly the same, but they don't look exactly the same. So all those physical differences you're seeing, those are all evidences that are coming from the environment. And as you may be able to surmise, the twin that's the smaller of the two twins, this was the one that unfortunately went through these traumatic experiences and did not have that great environment growing up. So environment is really important. Your textbook gives us this great picture. I love the idea that people decide to wear this out in public. But secondly, I love this picture because I want you to imagine for a second that shirt color is inheritable. So looking at this, you've got daddy shirt and mommy shirt, and now we've got baby shirt. What type of inheritance pattern do you think is being expressed in this particular photo? Hopefully, as you looked at this, you realize it's not complete dominance, right? One of the shirts is not completely dominant over the other. This is actually an example of co-dominance, right? Where we've got both the horizontal stripes and the vertical stripes being expressed in the child. Again, shirts are not inheritable, just a fun way to review that inheritance pattern. Your textbook does a great job of going through these inheritance patterns and providing details and examples. I encourage you to go through those. You can also practice genetics problems. There are practice tips in your textbook. There are good practice problems in the text. There are also a lot of practice problems online in the study area that you can use. All of the activities use these little Mendelians. It's their cute idea, but these are great ways to practice doing genetics problems. To be reminded, you have the following types of problems guaranteed on your final exam: a monohybrid cross, a test cross, a dihybrid cross, and a multi-character cross. The other types of inheritance patterns you should be able to recognize from examples, sort of like we just did with those shirts on the family. We now want to skip up to chapter 15 and just look at concept 15.5 to look at some inheritance patterns that are exceptions to normal standard Mendelian inheritance. These are not going to be able to be determined by a Punnett square or using what we know about dominant and recessive. So one of the exceptions is going to be with genes that are located in the nucleus. This is called genomic imprinting. The second exception is going to be involved with genes that are located outside of the nucleus. Remember, most of our DNA is inside the nucleus, but some of our DNA is in our mitochondria. If we're in our plant, it's in our mitochondria and our chloroplast. So we're going to look at these two exceptions to normal Mendelian genetics for genes located in the nucleus and for genes located outside the nucleus. Let's begin by looking at genomic imprinting. In genomic imprinting, what happens is certain genes get stamped with an imprint during gamete production. What you look like or the phenotype of the individual just simply depends on which parent passed along the allele. No longer does dominant and recessive factor in. This is a very small fraction of genes that this happens to, and these are usually early embryonic type genes. We're going to look at an example on the next slide of a gene that's a growth gene. If you get the dominant allele, you are going to be normal sized. If you get the recessive allele, you're dwarf sized. And we're going to sort of look how genomic imprinting plays out. So here we go. Up here at the top, we get this mouse that has the normal IGF2 allele from dad and the normal IGF2 allele from mom. And so we might write that this individual is homozygous dominant. So they're normal size. That makes sense. Let's go to example B. This is going to get the normal IGF allele from dad and the mutant IGF2 allele from mom. So we might write this individual as being big I, little I. Oh, they're normal size. That makes sense. Let's look at this third case. In this third case, they get the recessive allele from their father, so little I, and then they get the normal IGF2 allele from their mom. Normal Mendelian genetics would say, oh, they have a copy of that normal allele, they're going to be normal sized. But we don't see that happening. What happens because of genomic imprinting is no matter what the mom gave the mouse, it doesn't matter. That DNA has been blocked. So in this case, that normal gene that mom gave has been locked. So this mouse is going to be dwarf in size. Again, note that we can't really use normal dominant recessive anymore to talk about this. We're just looking at which parent gave this particular gene. Imprinting is the result of methylation, the result of adding methyl groups. Oh, there it is, one of our functional groups. Right? Adding methyl groups to DNA can cause this DNA to be turned off in these situations. This really only affects a small fraction of mammalian genes, but these are really important genes for embryonic development. Our second example that we want to talk about is the inheritance of organelle genes. Mitochondria and chloroplasts have their own DNA. It's not in the nucleus. And so what happens is this DNA usually gets inherited maternally. If we think very simplistically, here's a picture of an egg. That egg has the DNA in it, but it also has all the organelles. So it has the mitochondria and the Golgi apparatus and the ER. The sperm, on the other hand, is very small. And so the sperm only has room in it for DNA. There's no room for anything else. And so what happens is you normally are going to inherit your mitochondrial DNA from your mother. This can be implicated in various diseases such as mitochondrial myopathy and Leber's hereditary optic neuropathy, where it's not inherited in a simple dominant recessive in fashion. It's did mom give you this gene, right? Because mom gave you this DNA in their mitochondria. So this gets us to the end of chapter 16 and sort of wrapping up chapter 14 and wrapping up the course. As we think about biology and what we've done this semester, what you'll probably remember a year or two from now, hopefully you'll remember that Gwen Christine is your teacher, right? That was good stuff. GC equals TA. Maybe you'll remember some funny mnemonics. Did King Philip come over from Germany stoned? Ways that you memorize stuff. Maybe PP Matt, Sean. You'll definitely remember the coronavirus was going on. In reality, though, I'm hoping that maybe you'll remember some other biology stuff. I really hope that we've talked a lot about what it means to be alive and what it means to study life, and that you're confident with understanding what it means to be alive. We've spent a lot of time comparing and contrasting prokaryotic and eukaryotic cells, and you've gotten really good at doing that over the course of the semester. Global warming is real. Increases in carbon dioxide are real. You have a real you should feel motivated to make a change in how you live and to help others make changes as well. Hopefully you recognize that vaccines are good, that tanning is bad, and I hope that you understand how the process of CRISPR works. This is a technology that you are going to continue to hear more and more and more about, this way to edit DNA, and I think that it's something that you're going to want to be able to make an informed decision about as you continue. Really, what do I hope that you remember most about biology is that I hope that you've learned some critical thinking skills about how to make decisions, how to evaluate data, how to look at data, how to understand science, and make good decisions as an educated adult. I've had a great time this semester. I've missed seeing you for this last little bit of the course, but I'm really excited to see where your college careers take you and what you continue to do with biology. As you continue on, definitely feel free to reach out to me if you've got questions or as you go through future courses. I would love to see and hear from you as you continue. And that